I did my presentation on polymerase chain reaction, or abbreviated as PCR. My introduction. PCR is used to make billions of one segment of DNA out of the whole strand of DNA that you're working with. And that just means that instead of taking a long time to get one segment of DNA, you can take it and make a lot of it at one time. And the purpose of this lab was to determine who committed a crime, and we had DNA that was collected at a crime scene to compare with all of the suspect's DNA, and the sample that most closely matches the crime scene DNA is the guilty person after we run it. My procedure, we used amplification of DNA using PCR, and the steps to that is the initial denaturization, which takes the whole double helix DNA and splits it into two parts, and then it goes through 35 cycles of denaturization, annealing, which uses the primer that we added to it, and it attaches to one of the strands of DNA, and then the extension, which copies the entire strand. And then it goes through denaturization again, 35 cycles of just splitting, attaching, and copying. And then the final extension, which is just the last step, and it leaves you with a billion of the right segments of DNA that you want, not the whole strand. And it only leaves a small margin for error that way. And then what we did was we separated the DNA using the gel electrophoresis, which we ran, and then we compared the bands of DNA to how far they were, and the marker row, which was the crime scene DNA, to determine a match. And I would have taken a picture, but our sample was kind of iffy. We couldn't take a good picture of it, but we could see it fine, so I made a replica of it. And we can see that the crime scene DNA has 4 bands that all match up to suspect 2, but suspect 1 has a lot of different ones. And we perform PCR to create enough fragments to make a good match to it, because like I said before, we can just make a whole bunch of the right kinds of DNA at once instead of using the whole strand and possibly making a mistake. And this is the alternative to getting cells and then growing them and introducing the DNA for them to copy, which takes a very long time. So with this, we could do it in maybe 30 minutes or so. And our data showed that suspect 2's DNA was a match to the one collected at the crime scene. And we know this because the DNA markers that ended up on the electrophoresis were the same molecular weight for both of them, which just means they traveled the same length and matched up. Our room for improvement was to use a liquid ethidium bromide stain, kind of dangerous, but that's the best one to use because we used an ethidium bromide stain patch, and that didn't stain it enough, so we couldn't take a picture of it then, and it would just work out better. And we would also want to heat fix the stain next time, so we could like leave it overnight or move it without the stain possibly being contaminated. And we'd like to thank the Toshiba America Foundation for providing the funds, and Mr. Lee and Mrs. Hutson for all the help they gave us.